Let's move on to the first plenary session of this wonderful conference. And it's a pleasure for me and an honor for us, the organizing committee and the University of Évora, to have among us a colleague, a friend, and one of the most, I would say, rewarding researchers in Portugal, the young generation of research. Jaime Ramos from the University of Coimbra, which is actually the oldest university in Portugal. And this is quite interesting because, as you probably know, University of Évora is the second oldest, although there are some differences, but nevertheless. Jaime is working a lot with birds, not just with birds, main fields of conservation biology, and he enthusiastically accepts our invitation to be the national guest for this conference. So, with no more words, I will leave you with Jaime. Please, Jaime. Thank you.
Thank you. Okay, so good afternoon. First of all, I would like to thank João and the organizers of this conference for inviting me. As you know, there are very good ornithologists in Portugal, so I hope that by the end of this talk, you think they made a wise decision by inviting me. And I was just talking to my friend Ivan, he's around somewhere, where I was there. He said, if you talk more than 20 minutes, no one will pay attention. So I will be talking more than 20 minutes, but what I will tell you basically 3 stories. So I hope that each story gives a kind of intermission for you to kind of relax and go into the next story, okay? So this was the approach I took. I work now at the University of Coimbra. So basically, I could start with this question. When João invited me, I said, what am I going to talk about? So I thought, well, how did I begin? So basically, I began, like most of you in this room, by counting birds. And when we count birds, well, in my case, my undergraduate project was about the abundance and distribution of the purple gallinule in the Algarve. So when you're counting birds, in fact, you are addressing a very interesting question, which is the reasons behind changes in distribution and abundance. So this is perhaps one of the most fundamental questions in ecology, and everyone in this room really kind of is motivated to this question. But as we move, along our careers, along our jobs, we meet lots of people and then we kind of switch and change, but we always keep this kind of basic question in the background. So basically, taking this into account, I decide to take this approach and decide to give this talk, which is basically a compilation of several stories using different bird species and addressing this fact that understanding the drivers behind populations is a very important thing. And of course, then as you will see in each of the examples I will show you from the studies I've carried out, not just myself, but myself and people that have worked with me, we moved a little bit to community ecology and using birds also as indicators of environmental change, and we think this is an important step that make us understand better nature and communicate with the public. And the species, the birds that I will show you today will be the Azores bullfinch that was briefly mentioned before, and then there will be another story about the little tern / roseate tern, and then the Corey shearwater. So these are really kind of very specific species and the stories that I will talk you about, which may be a little bit different from other things that this audience does. So now we decide to call ourselves, our group, the Ecology and Conservation of Top Predators, and we call ourselves EcoTop. Sorry, I go back here. And basically, I have this picture here to show you several people. Of course, all the data I'm going to show you, I didn't collect it myself, but some people like Vitor, Catarina, Joana, and many other people. have helped, of course, collecting data for their thesis and for the work we've been doing. And also I've been also collaborating with several organizations like SPEA, and some of the data was collected in collaboration with this organization. So the first story is the Azores bullfinch. You may have heard about this species. It's a very specific species, endemic from the Azores Island, with a very restricted distribution, and is endemic. It's endemic to this island, São Miguel, which is 2,000 kilometers west of Lisbon. But if you go to São Miguel, it's a very nice place to go. You have to visit this area here, which is the SPA of Pico da Vara, Ribeira do Guilherme, where this species exists. So it's a very restricted species, and when I basically began working with this species was in 1991, and when I basically landed in São Miguel, the only information I had was from 2 previous visits that Colin Beebe made to the area, and he told me, oh, you'll have around 100 pairs of this species breeding in the area. So you go there and we have to find out what he eats, what he feeds, and things like that. So basically, I was just dropped there on my own. And of course, the first thing, as I told you, we start with bird numbers because this is like the basics. And of course, doing census is always not easy, as you may know, but in this kind of habitat is even more difficult, very hilly, dense vegetation, rains a lot. So what I had to do in the 1990s, basically I relied on lots of tracks in the area. Some of them are numbered here. And I just marked point counts every 200 meters. And in those point counts, I stayed 8 minutes in order to estimate the abundance of this species. Of course, immediately you know, of course, but these point counts are not randomly distributed in the area. There will be lots of problems. Yes, of course, we knew that, but that was the only way I could access the area. Later on, within the umbrella of as Domingos mentioned, within the umbrella of the Priolo census that we developed when I was also connected with the project, the things moved a little bit better because a grid was superimposed on the area, and 307 points were marked in the corner of each 1 by 1 kilometer and in the center. And all these points were surveyed by 48 people in one day. So this was quite an achievement. So this will happen again next year, if you want to join. It's lots of fun. But of course, this was a much better design. I have to tell you, of course, that some of these points were not surveyed at the exact location because of safety reasons, because that was not possible. But anyway, having all this information, I can show you this graph with the bird numbers of this species. So basically, we know that it was a common species by historical chronicles in the 16th century, and then must have been common in the 19th because it was a pest in orange crops, feeding on the flower buds. By the 1970s, some ornithologists in the Azores estimated around 40 pairs, and then Colin Beebe here 100 pairs. When I was there, because I rang birds, I did some capture-recapture. I estimated around 120 pairs. This capture-recapture was without modeling. And then here 2005-07, we rang more birds, and then here we were able to model the population with the program mark, and the estimation was around 800 pairs. And then during the census, during the Priolo census, things decreased a little bit, but to nearly 600, and here slightly less. But anyway, so of course this graph shows two things. One thing is that there are really wide bars. I mean, like here, this is the average, but of course we did several models for several years, and there is a huge variation, as you can see. It's not so easy to estimate precisely the population of this species. And the other aspect, this is for, basically works out for the whole methods. And the other aspect that is important is that there was apparently a jump, so the bird increased enormously after 2005. Of course, because the habitat restoration work, the conservation work had begun in 2003, we would like to think that possibly this was due to some habitat restoration work, but also possibly because there was some changes in the behavior of the bird because it could not change so much in such a short time. Or maybe we were doing something wrong and before we are not covering the area very well. But anyway, with this method, we were able to estimate the extent of occurrence, and the bird was downgraded from critically endangered to endangered. The other thing that we worked a lot in this species was the diet. The diet of this species is a very peculiar species. I mean, each species is peculiar in itself. But in this case, I mean, it feeds on many plant species. I'm not going into detail, but it varies every month as the new plants come into fruit. But I would like to highlight two aspects here. The first is that it feeds a lot on some exotic species, for instance, Cletra here, which is an exotic species in San Miguel, and also feeds a lot on ferns. These are 3 fern species, Uduardia, Culcita, and Osmunda. Throughout the winter, it feeds a lot on ferns, which is quite uncommon for birds in a way, or at least you have to be in a very wet and humid climate. And of course, this other picture also shows that really the bird feeds a lot on both native and exotic species. So here is the percentage of foraging observations, and throughout the year, it takes both native and exotic species, because there is a huge and aggressive invasion of exotic plants in the Azores. So, I mean, as we moved on and carry on our work, there were 2 main ecological questions that kind of emerged from these first studies. The first was this strong biological invasion. I mean, what could be the effect on the bird and the biodiversity of this ecosystem? And the other aspect, which is a bit peculiar but nevertheless very interesting to this species, is this aspect of fern feeding, which is not that common in many species. So going on to the first aspect, basically, and this was basically the work that of a PhD student, Ruben Ilen, when he was there, we addressed this biological invasion. And of course, addressing this biological invasion, the question is, when you have invasive plants and these increase, like the Cletra and other plants, of course you expect a decline in native species, a decline in insect species. Of course, the birds like the bullfinch would possibly also decline, and the ecosystem becomes more homogeneous and also suffers some biological invasion. If we do the opposite, meaning if we are able to remove the exotics, I mean, the native plant species should increase, the insects, the birds, and the whole ecosystem would benefit. So the way that we planned for Ruben to address this was, we decided to address these using 35 transects along a gradient of invasion that we established in the study area, which I show here in yellow. Let's say these transects, these sampling plots were 30 meters by 5 meters, and in those plots, all the seeds were removed, and we use as the measure of insects all the insects that emerge from the seeds. So basically, this is our design. We have here our 35 plots along a gradient of invasion, from some plots really composed of only native seeds, and the other ones highly invaded, only composed with exotic seeds. And this is the mean number of exotic seeds. And then we try to assess what were the implications in several biodiversity descriptors of this increase in exotic seeds. And what we got was, so basically, the invasive plants reduced the seed abundance from native species, obviously. So here you have seed abundance from native species, and here in the x-axis, the gradient of invasion. Of course, the insect species richness was also affected, but here we had a quadratic relationship, meaning some with little invasion, there was some benefit on the species richness of insects, but of course, with a high invasion, then there was a steep decline. The insect biomass also decreased because we were able to weigh these insects, and the evenness of the insects also showed a quadratic relationship like the insect species richness. So basically here we are showing that this strong invasion of the ecosystem were having very strong effects on several biodiversity descriptors that we're able to assess in this one season. Of course, if we do, let's say if we think, if this is our invasion, most invaded, less invaded, if when we do restoration, we would like to basically move to this side, so we would like to have the opposite of this graph, and our question was, will the control of invasive plant species result in the correct restoration of the ecosystem? That was our question. And this, of course, this relates a lot with with the LIFE project that the SPEA, the Portuguese BirdLife partner, and other partners were carrying on in the area. And we kind of joined to collect data. So basically, I'm going to show you only a way of addressing that. We had more data, of course, but basically we had a control plot composed mainly of native plant species. Only about 2% of exotics, because we couldn't get only natives. And we had a restored plot that was restored by the working team, and all the exotics were removed. So this is in year 0. These ecological network graphs, as you may know, I mean, here we have the birds, the seeds, the insects, and the parasitoids that emerge from these insects. And these seeds were collected from the birds that we were misnetting. And of course, each cell here represents one species, and the larger the cell, the more important that species here. As you can see in year 0, the control area had some very little aliens here. The restored area had many more aliens. And this was, I'm not going to much detail, but this was the links among these, sorry, I'm going one back here. The links among the several trophic levels. We didn't sample the control plot in year 1, but we did sample in year 2. And as you can see, remain more or less the same. There was some increase in the alien plants here. I mean, these bars here increased a little bit. But the restored plant, the restored plot we sampled in year 1. Of course, there was a decrease here in the number of alien seeds because the alien plants were removed. And in year 2, you can see that there was really a large increase in all these cells for the seeds, and all in here in the cells for the insects. So really, when we removed the alien plants, we were improving a lot in terms of abundance and species, the biodiversity for the native seeds and for the insects, because I mean all these here increased a lot. Whereas when we compare year 0 with year 2 for the control plot, there was basically no difference. But of course, there was also a drawback here, which is that, as you can see here, I mean, we removed the alien plants, but then they were kind of coming back in year 2. So somehow, of course, they could be coming from other areas. They could be coming from, flown in by birds from nearby areas. So basically, although we were certainly improving the biodiversity of the area, I mean, there was problems. And the other problem that we thought would be that with another invasive in the area, which is the black rats. So we asked this question: does nest predation by the black rats could be increasing as we were restoring the habitat? This question makes sense because if we restore the habitat, as I told you before, we are increasing the abundance of seeds and the abundance of insects. And of course, the rats, they feed on seeds, insects, they are generalists, they feed on earthworms. And so in order to address this, in collaboration with the SPEA team in São Miguel, we have been using quail artificial nests that we placed to see during 20 days if they were predated or not. So basically, we did this in two areas, one that we called successful restored area, which is here in black, and the other one we called partially restored area, that is here in gray. Let's concentrate on the black area. So year 0 was 2008, and year 10 was 2018, last year. And as you can see, from year 0 to year 10 after restoration, there was a strong increase in the predation of our artificial nests. So, and this was significant after year 8. So, of course, why was this? I mean, possibly because the rats were increasing in abundance, and we had estimated them with snap traps in 2008. We did again the same last year, and really there was a huge increase in these 10 years. So although we are restoring the habitat, we were improving the habitat for the bullfinch and for the rest, I mean, there were things that we are not able or we're not doing well. The other point about the bullfinch I told you that interested us a lot was this thing about the fern feeding of the bullfinch. So basically, I'm not a botanist, but because I kind of got involved with this, I mean, this bird feeds on ferns, not many birds feed on ferns. Is this a common thing in other areas where you have many fern species, such as New Zealand, whatever? And basically, I met a pterodologist from the University of Madrid, Luis Quintanilla, that is now my friend, and he kind of even invited me last year to give a talk at a symposium of cryptogamic botany, and which is very unusual for an ornithologist to go to a symposium of cryptogamic botany. If you know what is cryptogams, I'm sure you do. So basically, in that talk, I try to convince these cryptogam botanists that vertebrates contribute to short and long distance dispersal of cryptogams, not just ferns, but other cryptogams. There are some species that their ecology is very much related and linked with ferns, such as the Azores bullfinch. And they were not really considering cryptogam dispersal by animals, but they should, because if they consider them, they could help explaining the distribution of many ferns and other cryptogams, such as mosses and other things. So basically, what have we done in relation to this other aspect of the ferns, of the fern feeding ecology of the bullfinch? Well, in the things I did in the 1991 when I was there, as you can see, this is the percentage of the foraging observation on ferns, and the bullfinch feeds a lot on ferns in winter, from November till June, but really is a winter feeding thing. So later, I mean, like, we should consider, I should give you this information, that there are approximately 71 classified fern species in the Azores, so this is a huge resource. I mean, the birds, they feed on spores, and they leave these big marks here that are very easy to identify, because no other bird in the area, so if you go there and if you see these, if you go to San Miguel, you know there was a bullfinch feeding on these ferns, because no other bird has this behavior. But they also feed on the fronds, I mean the leaves of the spores, such as this royal fern, and they leave again conspicuous big marks. So, taking this information that I had from the 90s, we decided that these were the two most important species, the Woodwardia and Culsita, we decided to do this again, basically, and we went back to our transects now that we knew the area quite well, and we ran 259 transects to evaluate the abundance by the number of fronds, meaning leaves that have the spores, and consumption by counting the number of leaves that had these big marks that we could identify very well. And the conclusions kind of they reached what we thought would be. So basically, this is the abundance, the number of leaves here of the different species. We did this in different altitudes. Of course, the bullfinches, they were not feeding in relation to the abundance of the fern species. As you can see, for instance, here, Dryopteris is the second most abundant, but they basically did not feed on Dryopteris. So they took Woodwardia and Culsita, as I had observed previously, and they also took another species, the Pteris. So basically, our next question was, why are they doing this? Why are they selecting certain fern species and not the others? And of course, I mean, the most immediate thing is like, this must be related with nutrition, because this is a winter feeding behavior. This is quite a cold, humid, damp area. The birds need energy in winter. So of course, I mean, talking with Luis and several other people that know more about nutrition of ferns, they immediately told us, look, the spores of the ferns are very rich in lipids, so you should go and look for them. So that's what we did. These are the lipid drops in the ferns. And we examined 3 nutrients, lipids, proteins, and the calorific content. And basically, the only one that differed between the ferns that were used by the birds and the ferns that were not used was the lipids. So the lipids, really, the birds were selecting the spores from the species that had higher lipid content, which makes sense because they really need this energy in winter. Anyway, so this was like, I started from just counting the bullfinch, and then I was like doing work with cryptogam biologists. And anyway, so I found this thing quite exciting. And of course, if you go to the literature, I mean, there is a new move about birds as mobile links. Birds are very important in moving things around. And so we should consider them as very important in moving things around. And of course, the other aspect that immediately we thought, or at least Luis told us, look, the birds could also be very important as agents of dispersal of the ferns. So we took fecal samples and we took them to Madrid, and Luis germinated them in the lab. So this is the number of fecal samples for each species. This is the number with viable spores. So many of them had viable spores, particularly the Woodwardia that the birds prefer the most. So they were really dispersing the ferns. So they were very important as dispersal agents. Of course, you may say, oh, but this does not make much sense anyway. They could disperse, but as we know, I mean, wind dispersal, it's a very important thing for ferns. But we can also say that the quality of dispersal by the birds may be much, much higher than the wind dispersal, because they disperse the ferns in the dropping, so the likelihood of them germinating should be much higher. At least in the lab, they germinated very well, and we proved that the bullfinch were really acting as fern dispersers. I mean, the whole idea as mobile links, and starting with this, also drove us to another work that I'm only showing a graph, that was, again, part of the PhD of another student. And we did this in Coimbra. So basically, we thought that even the birds in Europe, they could be important as pollinators, or at least as mobile links of pollen of plants and other things. So what we did, this was done in central Portugal. What Luis did was we sampled lots of birds throughout the year. So basically these are the total number, and this is the number of the birds that had pollen in their forehead feathers. So basically to do this work, we just contact a pollinologist in Coimbra that is able to identify all pollens. And so we just gave him the feathers, and he was able to identify the pollen for us. And we could show that the birds in central Portugal could be even important as pollinators in midwinter. I mean, they were transporting lots of pollen. And not only they were transporting pollen, but they were also transporting fungi along with the pollen. And of course, I mean, and then this could lead us to several other things. I mean, these fungi could basically have negative interactions with when you use deposit on other flowers, or positive or whatever. But anyway, the thing is, I mean, we did this work in central Portugal. I mean, here in Europe, as we are all ornithologists here, we don't have any specialist nectivores in Europe, basically. I mean, but if you think that in tropical areas with so many specialist nectivores, I mean, we should think that not only pollen, they are not just moving pollen, but also fungi and other things. Anyway, we found this quite interesting, all these interactions among the different levels of biodiversity. So this is here where you relax. I don't know if I reached the 20 minutes, but anyway, I'm going now to switch to another story. This other story is a bit different. I moved away from this, from the bullfinch to another bird that I've been working with, which is the little tern. So I'm going to show you some aspects again along this same theme of the work with little terns that we've been doing in the Algarve. So the Algarve is here in southern Portugal, where most people go on holidays. But this is the Rio Formosa Natural Park. You have like these sand barrier islands. They communicate to the sea by these channels. And the birds, the little terns, they breed on these islands. And they also breed on salinas, mostly Tavira and Olhão. As you probably all know that, I mean, we're talking now about a bird that feeds mostly on small fish, that he captures mostly in the lagoon. And when we start doing this, we were interested in two things in terms of bird counts. We were interested in the numbers at the regional scale, I mean at the whole scale of the Rio Formosa, but also at the colony scale. I mean, we would like to know how each colony, what would be the beginning and end in terms of bird numbers of each colony. So these are the two scales we were working. So the data, we used some data from that several Portuguese ornithologists did census between 79 and 93. The only data we got in these reports was just number of breeding birds and vegetation cover from aerial photos that we obtained. And we have been working since 2002 on this species. And basically we go there every year. I mean, it's very nice. We do this in May and early June. And we assess the number of breeding pairs. We assess the vegetation cover using aerial photos and in situ. And we have data on reproductive parameters, mostly clutch size, egg size. Not breeding success is a bit difficult. We only have data for a few years. We have data on diet, that we identify otoliths and fish scales from the pellets, and we also have data on environmental variables that explain the food resources, I mean the fish that the birds feed. So let's go to the bird numbers. At the regional scale, this is the census of the little terns. As you can see, I've divided, these are the salinas in dark and the sandy beaches in gray. As you can see, most birds prefer to nest on sandy beaches. But the thing, as you may see, there are huge variation in the number of breeding birds every year. So our first question is, what is driving this annual variation in the breeding population? The second question was, what is driving the number of birds breeding in a certain colony? For instance, this is the colony of Vermona that began in 2006 with 5 breeding pairs, and then increased, and then last year there were none breeding there. So again, the question is, what was determining the beginning and end of each colony, the two different scales? Of course, to address the first question, we thought that, food resources would be the most likely driving factor. We had some data on the abundance of fish, but not so many, but in the end we used some surrogates. And one thing we knew from the diet is that these fish species, sand smelts, they are the most important species in the diet. So every year they constitute from 50 to 75% of the diet. And this graph shows the importance of the sand smelts in the diet in relation to the mean sea surface temperature in February. As you can see, lower temperatures in February, which is when the sand smelts spawn, lead to the sand smelts being more important in the diet of the little terns. And if you go to if you talk with fishery biologists, these lower temperatures are also important for the spawning and the survival of the eggs and the larvae, not just the sand smelts, but sardines and other fish. So the birds were really, we could show that the birds were really reacting to these. For instance, the clutch size increased with the importance of sand smelts in the diet. So in years where the birds were eating more sand smelts that we had in our pellets, we could see that the number of eggs per clutch was much higher. And the breeding population, we tried different things. The breeding population was related with this difference in temperature. The breeding population was related with the difference in temperature between May and June. So the wider this difference in temperature between May and June, it seems that more birds would come into breeding condition. So this means that probably you would have quite low temperatures in May, and then you would have like a steep increase until June. Of course, here we use the mean values. And the birds apparently reacted to this increase, and when you didn't have any anomaly, it was the same for the 2 months, our breeding population was much smaller. We thought this would be interesting because some other work I have been doing, again with terns, very far away on Arid Island in Seychelles, we could show that the birds were really responding to this increase, in this case not in temperature, but what we showed was an increase in the chlorophyll concentration. I will explain this. So basically, this graph here for the terns, for the roseate terns on Arid shows this is the chlorophyll concentration each month, February, April, June, here. As you can show, there is a peak, as you can see here, there is a peak around June. So basically, this is when the birds breed. So the birds breed when there is a peak of food, as we all know. And what we did was, we did a regression between the chlorophyll concentration from the mean laying date, which was usually around May or sometimes early June, until the peak of the chlorophyll concentration, okay? So we did this regression, and we call this the rate of increase in chlorophyll concentration. So we thought this rate of increase would be related with the rate of availability of resources in the area, which would be likely small fish, of course. And this worked out very well because, for instance, here in these graphs, as you can see, in 1999, basically you have like the rate of increase was virtually zero here, and the success was zero. In 2004, for instance, I'm showing only some years of data, we had the highest increase in chlorophyll, and we had the highest success ever of all the years we have data. In 2002, we had 2 peaks in the chlorophyll, and we did our regression line was to the midpoint, but again, there was some increase and the birds had some success. And in 2005, the rate of increase was not so high, as you can see, was not so steep here as here, and the success was on average. So we thought, I mean, like for a bird in a subtropical area like in the Algarve, really needs this chlorophyll or this temperature kind of moving on and bringing the fish, so more birds to come into breeding condition. I mean, so we are more or less quite interested in that this could explain the breeding population at our regional scale. So let's go back to the little terns. So basically, let's go now to the local scale. This is our colony of Hormona here. The birds began breeding here in 2007. As you can see, by 2018, last year, all this area was covered in vegetation. And of course, those of you that are familiar with little terns and other birds know that they really avoid vegetation because vegetation is related with predation, where predators hide, and so on. And of course, this increase in vegetation, I mean, correlates well with our graph here of an increase in the number of breeding pairs of the breeding pairs in this area, and then a decline. So when we did this, using all the data we had from all the colonies in relation to vegetation cover, so here I show the percentage of the number of breeding pairs in our sandy beaches in relation to vegetation cover. As you can see, the highest number of breeding pairs is around 10% vegetation cover, but there is a break point here at around 42%, sorry. So from here to 42, the decline is not significant. But after 42% of vegetation cover in a certain colony, the birds start to decline significantly, and basically by around 90%, all the birds leave. They just abandon the area. So basically, at our two scales, we could see that environmental variability affecting food resources should be important, as we would expect, should explain the population size at the regional scale. But at the local scale, we could see that the overgrowth of vegetation, which I'm not going to show here, but was related with increase in predation, leads progressively to a decrease in colony size. This also leads to a decrease in the mobbing behavior of the birds, and the mobbing behavior is very important as anti-predator strategy. And this leads to a fugitive strategy, which is something that was already, this term has been coined by Joanna Berger in America. And the birds move from one area to another as a response to this. I mean, this aspect of the mobbing behavior for these shore birds is quite interesting. And also, when you look at these also motivate us to do another work that Afonso did in Samuco, in the Tagus estuary, which is very much related with this topic, which is he examined the probability of nest success of Kentish plovers in relation to the overlap proportion with the breeding season of the black-winged stilts. As you know, black-winged stilts are very noisy. They show a strong mobbing behavior. So basically here, using several years of data, we could show that the Kentish plovers were more successful when their season was overlapping with the season of the black-winged stilts. So I mean, all these, of course, what this means is that these multi-species colonies of shore birds are very important for these birds to be successful. This is not 20 minutes, Ivan, but it's like, I don't know, 12. So I'm going to my last story. This is like several stories for this first day. I hope you like it. So my last story is the Cory shearwater, which is another species that we have been working. Again, when I was working at the University of the Azores, of course, the first thing we do again is counting birds. So we went to work on an islet that was a reserve. This is Ilhéu da Vila, Villa Islet. And the first thing we did was a complete nest survey. Took us almost a month. And we estimated the breeding population of Villa Islet as 311 breeding pairs. This was in 1994. But of course, for all those of you that have been in a Cory shearwater colony, you see, I mean, there are so many birds arriving at night. You immediately wonder why. I mean, there should be more birds breeding here. Why aren't there more birds breeding here? And the obvious thing is that there are not more birds because there are not enough nest sites. I mean, this also is important for other species. So what we did in the following year, we built 28 artificial nests in February, and more or less half of them were immediately occupied in that same year. So basically, this suggests that the breeding population here was highly limited by nest site availability. So there was not enough nest sites for the population of Corys. We also thought about how to estimate bird numbers of Corys in the Azores. This is not an easy task. And together with Luís Monteiro, that sadly is not with us anymore, we decided to count birds in rafts. We did that close to Villa Islet, and we counted, this is a mean of several counts, 1,500 birds. I mean, many more than the birds we had breeding. We decided to take this approach to the whole archipelago, and we went like crazy, invited several people and counted rafts everywhere, and reached a figure of about 500,000 Corys for the whole archipelago. I mean, but in the end, we never published this data because, I mean, there are so many factors influence the raft numbers that in the end we never managed to. But anyway, it was a very interesting exercise. So basically, we moved away from these, again, bird numbers, kind of drift, and recently, and this is the last story to finish, we have been dealing about the use of Cory shearwater as sentinels of environmental change. So basically here the rationale is, I mean, you know, there is the marine environmental variability is out there in the ocean. It can be captured by climatic indices, such as here in our area, the North Atlantic Oscillation. I mean, in other areas, the El Nino or whatever. Of course, this variability is linked to oceanographic data, such as sea surface temperature and chlorophyll concentration. And, of course, the Corys, when you have a change in these, when you have a change in these indices, basically this is just a difference in pressure between the Azores and Iceland. There is a change in the sea surface temperature. There is a change in the fish in the sea, in the abundance, and the birds, being top predators, they should pick up all these changes. This is the rationale, as you may know. So basically, recently we have been working with new technologies using GPS loggers. The data I'm going to show here, I mean, we have many more data, but I'm just going to show here an example. I'm going to show you here the data for May, which is the female laying azodes, and for one colony in Berlenga Island, which is in the west coast of Portugal. For these years, I mean, we have also been doing every year work in September for the chick rearing period. So basically, we deploy the loggers to get the positions of the birds, and then we have measures of foraging effort, like trip duration and flying time. We have measures of fitness, such as body condition and hatching success. And of course, we have measures of the environment, such as these indices at the North Atlantic Oscillation Index, sea surface temperature, and the chlorophyll concentration. And I'm going to show you these female laying azodes data, mainly because when we look here, this is the variation of the now in winter, December, March. As you can see, it varies from positive to negative and brings more or less storms to Europe. But in 2010 was really an extreme. It was a very, very a negative value. And these, of course, had very strong effects in the ocean. So 2010 was the year with the lowest temperature. So this is our study colony here, Berlenga, north of Lisbon. So this is the sea surface temperature of these different years. But as you can see, in 2010 was really a very extreme value. Of course, when we go to the chlorophyll concentration, again, it varies, but I draw your attention, this is the mean, for the value in 2010, again, was very low. And we were lucky to work with the oceanographers, and there is a fishing cruise going along the coast of Portugal, mainly because to estimate the abundance of sardines. And they estimated, they did some extra work for us, and in 60 kilometers around our study colony, they estimated from acoustics the total number of small pelagic fishes, meaning mackerel, sardines, and others, and the sardines only. I mean, the sardines were basically collapsed in 2010. As you can see, the total small pelagics were up and down, but again, there was a minimum in 2010. So what did the birds do? Well, I show here several years. I mean, because these are around these extreme values. For instance, 2007, the birds, they forage here in this lion domain, which had, this is the primary productivity, which is had reasonable values of primary productivity. In 2008, they switched and foraged in this king's domain here, which had higher values of primary productivity. 2010, they went back again to this lion domain, this area here, which had high values of primary productivity. And in 2011, which was immediately after this extreme, the females went all the way to Canada, almost to the Newfoundland, and mostly forage in this altar domain. This was in May, prior to the laying season. I mean, of course, this has been the work of several people, but mostly this was including in the PhD of Vitor Paiva. And we could see that trip duration, of course, was much higher in 2011. Flying time was much higher, so there was a cost, of course. And that cost is really seen here, that the body condition was much lower. I'm not going into details how we measure it. And the hatching success that the birds were falling was, again, much lower in 2011. So basically, in my final story, we were able to show that there was a decrease in the abundance of pelagic fish. This increased the foraging range of the cories, increased the foraging time and trip duration. There was a change in diet, which I did not show here, but there was. And the birds decreased their body condition breeding success. So they were really responding to these changes, and we could consider them as good sentinels of environmental changes, at least in these extreme years. Okay? So basically, you're probably all tired, so I'm going to finish. So I hope you enjoy my 3 examples. So basically, I just summarize here several points. Of course, I mean, from all the work I've been doing, I always have this feeling that correcting meaningful estimates of bird numbers is not an easy task. Of course, technology these days has improved things a lot, but it's not easy. I think it is important to think carefully about the drivers behind changes in bird numbers. When we think carefully about those drivers, we will be able to make a very strong conservation measure, because the people will understand more the causes and consequences of the things. In several cases, we were able to show that the birds really, they are benefactor species, like the bullfinch. They were benefiting apparently the spores, and the birds, the fungi and the pollen or whatever. And the birds are also very important, as you know, that they are good indicators of environmental change, and there are many more the literature is very abundant in those examples. And also the other thing I found out in these years is that when I collaborated more with people from other fields, like the botanists, the cryptogram botanists, the palynologists, or the fishery biologists, They always provided me with complimentary views, and they were very helpful in progressing and doing other things. And thank you very much for. We have time for one question.
No. We have time for several questions. It's up to you, and you, of course. Okay, thank you so much. Please.
Any doubts, any things? Was very boring.
I have just an informative question. Those bullfinches foraging on the ferns, and you showed the graphs of the ingredients of that food, which was dominant fat. Is there an alternative source of food that they use during the winter, or do they solely feed on those ferns, and are they just fatting up? But for what?
Yeah, well, now it's true that now they mostly use seeds of exotic species in winter, mainly that cletera, and that is possibly a reason why they have increased. Because the seeds, as you know, they have higher calorific content than the ferns. But in the past, really the ferns, if you remove the exotics, the ferns are really the most important, should be one of the most important food supplies in winter. And of course, I mean, some of the seeds, such as the blackberries, they will last until midwinter, depending on annual variability. Some other seeds from other native species will do the same, but certainly the ferns should be very, very important. Because I mean, there were also some areas when we removed the exotics like the cletera, in those areas the main food were really the ferns. In some areas around 90% during January, February. So really the ferns were very important. And we could also see, I mean, we don't have that much data, but with our colorings, that the birds were kind of moving along the altitudinal gradient when you had to reach the areas where they had, that's what we think, where you had mature spores with these lipids. But I mean, and since then, now I have been aware that many more people are seeing this kind of fern feeding behavior in Hawaii, New Zealand, even in Asturias, in northern Spain. So it's like, of course, when the resource is there, the animals use it.
Hello, Jaime. Thank you very much for your interesting talk. Back again to the Azorian bullfinch. I found it very interesting what you showed about the invasive species. But what I did not see, and I was kind of hoping to see, is the relationship. You showed this gradient of invasion, and you showed it very well how like the insects abundance and biomass decrease and everything. But you did not show us the relationship with the bullfinch. Can you tell us a little bit about that, please? Would be great. Thank you.
I mean, the thing is like we did. Ruben did that in 1 year, basically. So once, if you want to have a snapshot of the biodiversity varying along a gradient, so you pick up seeds and insects because it's easy. The birds, in fact, if you remember the plots, the birds didn't vary that much. It's true that they improved a little bit. But they didn't vary that much. But having said that, it's also true that the bird community in the Azores, I mean, in general, not just the bullfinch. The bird, of course, the bullfinch is just part of the community. The community is very small. So we're talking about in the area, I don't know, 7, 8 common bird species. So, and they're all very generalists. We're talking about island species, very generalists. So in that respect, there was some improvement in terms of bird abundance in the restored areas, but I mean, we could not show that positively as with the insects or the seeds, no.
In the bird stripping, are they picking the vegetative material without the spores?
When they're looking for the spores, they take the spores, and if you look at the birds, they just they release, when you observe them, they release from the beak the vegetative part. But when they're feeding on the fronds, the leaves, they go for the very young parts of the leaves, the very young ones, and then they take the leaves. Like in that royal fern, they go for the leaves. But when they have spores, they prefer the spores, yes.
Any more questions, please?
No. He's scratching the neck.
Like to say
Okay,
thanks
thank you very much.
again
Thank you, João.
to Jaime and also to Vitor and to Domingos and to you all for your presence, of course. And just I would like just to highlight. Well, it's difficult to highlight what Jaime said because I would have to highlight everything, but which is obviously impossible. But I would like to highlight the last phrase on this slide, because actually in the end, everything we do, it's about communication. Communication among ourselves, among scientists, among people that actually is involved in the work we do, but also communication with people, with general people. So what you have been done, Jaime, and we share with us just a portion of the work you have been done, because I know it. It's quite amazing, and it's all about communication in the proper way. So thank you again, Jaime. Thank you all. And it was a very rewarding plenary session. And keep in mind that the conference just will begin tomorrow. So it's not bad for the beginning. Thank you.